Hello everyone, this is me Swanshu here, and today we'll be talking briefly about glycolysis. What is glycolysis? Glycolysis is simple in simple terms is the breakdown of the sugar. In as far as our human body is concerned, the most important sugar in our body is glucose, from which we can generate the energy. Now, this pathway was given by Embedin, Otto Meyerhoff, and J. Parnas, so it is also referred to as EMP pathway. Now, in the question, they can trick you by either using the word glycolysis or using the word EMP pathway. Keep in mind that these both are the same thing. Now, as we will go through the steps of glycolysis, we will realize that there is no need of oxygen in the pathway. So this pathway can occur even in anaerobic environment, and it is the only pathway which can occur in anaerobic environment. So glycolysis is the only source of energy for anaerobic organisms. As it is written in NCRT, in anaerobic organisms, it is the only process in respiration. That is, it is the only source of energy in anaerobic organisms. Now we will look through some steps in glycolysis. Number one, we have glucose. We have to break down the glucose into smaller fragments so that we can obtain energy from it. Now we all know that glucose is 6 carbon compound and it is an aldehyde compound. So let's draw a 6 carbon compound with an aldehyde at the end. I'm not going into details of the chemistry here, but we all know that glucose is aldehyde. The first step is we add a phosphate group to the glucose. The reason is very simple. If you remember that the cell membrane is negatively charged and phosphate is also negatively charged, PO4 negative. So in order to keep the glucose inside the cell and not let it go outside the cell after it's entered into the glycolysis, we have to first add the phosphate, because glucose can easily transport across the cell membrane if there is no phosphate. So we add the phosphate so that the glucose cannot cross the cell membrane, which is also negatively charged, and this is also negatively charged, so it cannot cross. So the first step is we add the phosphorus to the glucose. Now, anywhere where you add the phosphorus, you need to have phosphorus from somewhere. In this case, we are adding the phosphorus from ATP, which is adenosine triphosphate. There are three phosphates attached to the adenosine group, and it is being converted into adenosine diphosphate, which has two phosphates. One phosphate group has been added to the glucose. Now, whenever you use a phosphate and add it to any molecule or any substance, the enzyme used is kinase. Here the uh, here the molecule or the substrate is glucose, so the enzyme is called as hexose because it is a 6 carbon compound, hexokinase, or you can also call it as glucokinase. They are both different enzymes present in different parts of the body, but for you, now you don't need to go in much details. Now you only need to remember that whenever you are adding a phosphorus from an ATP to a substrate, it is called as kinase. The enzyme is called as kinase. Now the next important step is we have glucose 6-phosphate. We all know that aldehydes are not symmetrical compounds. So what we do is we convert it into fructose, which is a ketone body. So now we can do like this, and ketone are more symmetrical than aldehyde. In order to break something, we need to have symmetry, and to have symmetry, we need to convert aldehyde into ketone. So we convert glucose into fructose, and as they both are isomers, so the enzyme is isomerase, and the compound is phosphoglucose. That means a phosphorus group is attached to the glucose, and we are isomerase, and we are making it as an fructose. They both are isomers of each other, so the enzyme is named as phosphoglucoisomerase. Remember, because this is an isomerase, there is no gain of energy or there is no loss of energy. After we have fructose 6-phosphate, we need to keep in mind that the phosphate is attached only at a single point. In order to break anything, the symmetry is very important, as I said. So for symmetry, we will add another phosphorus at the first place. That will make it fructose 1,6 bisphosphate. Again here, the phosphorus group is coming from the ATP, which is adenosine triphosphate, and the enzyme used is kinase, but the substrate is different this time. The substrate is phosphofructose because there is a fructose with an attachment of phosphorus group. These three are the very important steps in glycolysis. Rest all steps are not so important, but these three are very important. Why? Because number one, there are two ATP required. And as earlier on we said that glycolysis is a process in which we are making energy, but here we are seeing that 2 ATP are required. So this is also known as activation phase, in which we are giving energy giving energy to the glucose so that we can further create energy from it. Now we have, and why it is also important, the third step is most important step because this is the rate limiting enzyme of the cycle. Fros- PFK, phosphofructokinase, are of two types, 1 and 2, but now you don't need to go in details. But you have to keep in mind that phosphofructokinase is the rate limiting step or rate limiting enzyme of this cycle. Now we have fructose 1,6 bisphosphate. It is a symmetrical compound, so now we can break it down into three carbon compounds with each having one phosphorus on 3 carbon. That will give us glyceraldehyde 3 phosphate or dihydroxyacetone 3 phosphate. These both are isomers of each other, so we can convert them into each other. This is not required in glycolysis, so we will assume that this is also converted into this, and now we have two molecules of glyceraldehyde 3 phosphate, and this is converted into this by triose phosphate isomerase. That means a 3 carbon compound having a phosphorus is converted into its isomer, dihydroxyacetone phosphate, glyceraldehyde 3 phosphate, and we have now two compounds of glyceraldehyde 3 phosphate, which is each a 3 carbon compound. That means we converted a 1 6 carbon compound into two 3 carbon compounds so far. Now this is again an aldehyde. We will convert it into phosphoglycerate by using a hydrogenase enzyme, and we will also see that here also we are giving a phosphorus, but this time the phosphorus is not coming from ATP, so we will not use the word kinase. Rather than another enzyme is used, and that is hydrogenase, and we are using the ATP from our body, that is inorganic phosphorus that is in our body, and also NAD is converted into NADH. You have to keep in mind that if the question says of aerobic respiration, then this NADH can further go into the ETC and generate more ATP. But in case of anaerobic respiration, this NADH won't go into the ETC because there is no ETC without oxygen. Now we have 1,3 bisphosphoglycerate. That means we have two phosphorus. One phosphorus was already there, one we have added, and now we have again two phosphorus. Now we will extract this phosphorus and generate ATP. Why we have written 2 ATP here? Because we have two molecules of bisphosphate glycerate, 1,3 bisphosphate glycerate. So we will generate we will remove one phosphorus from each molecule and we will generate 2 ATP. And the enzyme used again here is kinase. Why we are saying the enzyme kinase? Because this is a uh, reversible reaction, so it can go in either way. If you go in this direction, then it will be called kinase. So if you come in this direction, then also it will be called as kinase. So now so far we have generated 2 ATP, we have used 2 ATP, we have generated 2 NADH. So net gain is so far is 0 ATP. Now then let's see the next step. We have 3 phosphoglycerate, and now we will convert into 2 phosphoglycerate. Again the symmetry matters and other things matters. We are just mutating using the help of mutase. Rather than at the third position, we are converting it at the second position. Phosphorus in this was at third position. Now we have changed it to the second position so that it is more symmetrical, and the other enzyme enolase can work on it. Enolase is a very uh, interesting enzyme which you will learn in first year of medical school, that it is inhibited by certain group 17 compounds like fluoride. So while doing any glucose test for patient, you have to keep in mind that you have to inhibit the glycolysis. So for inhibiting the glycolysis, you just simply add fluoride into the test tube so that the enolase won't work. If the enolase won't work, the glycolysis will stop, and the glycolysis will stop, so you can have an accurate measure of glucose. Well, forget about that, but right now just remember that 2-phosphoglycerate it converted into phosphoenol pyruvate in our body by enolase. Now we have PEP. PEP is a is PEP. If you keep in mind, if you can remember, then you have to remember that it is the highest energy compound in our body. And yes, by that I mean that it has more energy than ATP. ATP is used for generating energy, but phosphoenol pyruvate is the highest energy compound in our body. Now we have PP phosphoenol pyruvate, which is converted by a pyruvate kinase into pyruvate, and the phosphorus we had is now removed in another form of ATP. Because we have two compounds here and two compounds of pyruvate, the again we have 2 ATP. So now the net gain is 2 ATP, 2 NADH. But this pyruvate is then converted into lactate using NADH into NAD plus. So this NADH and NAD plus are nullified. If you see here that 2 NAD plus is made into 2 NADH, and here 2 NAD is made into 2 NAD plus because the compounds are 2. So, in summary, if we say by glycolysis we had converted glucose into pyruvate, or you can say lactate. If anaerobic respiration, the pyruvate is converted into lactate. If the oxygen is available, the pyruvate goes into Krebs cycle, and the further NADH, ATP, and FADH convert, which is further goes into the ETC cycle to make your ATP. But if it is an anaerobic cycle and there is no oxygen available, the pyruvate is converted into lactate. So you can say that in anaerobic phase for glycolysis, a glucose is converted into lactate with a net gain of 2 ATP. There is no net gain of NADH, as this NADH which was made here is consumed here, but there is a net gain of 2 ATP. This is all basic funda about glycolysis. If there is anyone who wants to know more about glycolysis, there are many things about glycolysis which you can't even imagine. But for you, for 11th students and for people appearing into NEET, this is enough for now. And thank you for watching the video. If you have any doubts or anything, you can definitely leave in the comment box. And if you would also like me to make a video on any other topic, you are always welcome to write in the comment box. Thank you and have a good day.